Welcome to this seventh webinar of Eufresco basic projects. And most of you already followed the previous one, but in case you didn't, there is the YouTube account where there are all the previous presentations. You can have a look and you can get further information about the basic substances and the potential basic substances. Three eminent speakers today will share their knowledge about the trials that are running in their lab. But before to start, I leave the floor to the coordinator of Eufresco, that is Baldissera Giovanni.
Thank you very much, Gianfranco. Good morning, good afternoon, and good evening, dear colleagues. I'm delighted to participate in this webinar. It has become a tradition, and I would like to thank Gianfranco and all of you for teaching us about this very important topic. Thank you very much.
Thank you, Baldissera. Your support is very kind and very welcome because we want to know and to share knowledge, and Eufresco Net is an excellent opportunity of interaction. And Eufresco is within HEPO, and HEPO in Europe is the basilar for the sharing of information and setting up of protocols for plant protection. And here we have Eva Matsjavic, and I'm sure I didn't pronounce well your last name, but I leave you the floor for a welcome and for a short presentation about that.
Thank you very much, Gianfranco, for this kind introduction. Indeed, my name is sometimes difficult to pronounce for people from well, different countries. My name is Eva Matyjaszczyk, but my first name is already usually easy to pronounce, so I always encourage people to use my first name. So I would like to welcome you on this seventh Eufresco Basics webinar, Potential Basic Substances for the Control of Downy Mildews and Post-harvest Decay of Tropical Fruit. And by the way of introduction, I would like to give you a very short presentation on what EPO is and why we involved in a project like BASIC. Sorry, I'm not sure how to move the screen. Yes, pardon. No, I went too far. So EPO is an abbreviation, and it stands for European and Mediterranean Plant Protection Organization. We are an intergovernmental organization created in 1951. So if you count years, we are already more than 70 years old. EPO was created by 15 member countries. Now we have 52 member countries, and on the picture on this slide you can see in dark green color the member countries of EPO. And while seeing this picture, we can see that EPO are all EU member countries, but also other countries from European Union, some countries from Northern Africa, and also some countries from Western Asia. EPO works in international cooperation in plant health. We are an international organization, and within the member countries, EPO works for and with national plant protection organization. What does it mean? It means that in every member country, our contact point, we contact with a governmental plant protection organization. EPO has two main areas of work. One is plant protection products, and I am leading the work on plant protection product. The second area of EPO work is plant quarantine, or preventing introduction and spread of harmful pests. As I work on plant protection products, and also plant protection products are relevant for this meeting, I would like also to stress that within area of plant protection products, EPO produces standards on plant protection products efficacy evaluation. And especially for participants of this meeting, I would like to tell that this year we are working on 3 standards on tropical fruit, on pesticides assessment used in tropical fruit. Hopefully this standard will be available for users in the autumn if they are approved by Council. We also have resistance database. This is already open for public, although not all data already are available. We are now filling the database with data from different member countries. We work on harmonization of data on plant protection products, mainly by providing EPO codes which are used in different databases. We have minor uses extrapolation tables and database which will include this minor uses tables is under development. And also we organize workshop in the area of plant protection products. So now the question is, as we are not a scientific organization, we are international organization working with governments, why we are supporting this project? And there are at least 3 good reasons for that. So basic substances are substances widely available in the whole EPO region, and they are just easily obtainable for farmers. They are not primarily used in plant protection, and basically there are no interested parties for whom promoting use of basic substances raises commercial interests. As you know, plant protection products belong to companies, and after developing and registering plant protection products, a company, industry makes a campaign of promotion and informs farmers and other users about the value of some products. Behind the basic substances, there are no industry. So information about their efficacy may be less promoted than about plant protection products. And since there are no commercial interests, it's up to a government, maybe scientists, and also international organizations and like ours, and maybe for other non-profit organizations to promote this knowledge. And last but not least, our superior body, EPO Council, recommended EPO to work more on IPM, and basic substances, in my view, are quite important for integrated pest management. So with this, I would like to thank you for joining this project and to stress that collaboration is very important. EPO succeeds only through collaboration between experts in the region and beyond. Thank you very much.
Thank you, Eva, for the excellent overview that you provided. The people that work within EU, and also I think the people that are outside but follow more or less what is going on this side of the globe, knows that EU planned to decrease of half amount of synthetic pesticides starting from the most toxic one in the next decade. That is not a decade, but is within 2010, 2030, so in less than 10 years. And this is not easy because we know that population grows, want to eat more and better, and so when we go to the retailer, we want perfect products. But so we need to spray more and more with less toxic compounds, and these basic substances can complement the synthetic pesticide. But as well, they are not well known, as already Eva told, are not promoted as other synthetic pesticides. Then not all times provide, we have to tell clearly, the same level of protection. So we need to integrate the different strategies, including those basic substances that are out of the rule. It means that they have their own rules. We don't need to register a treatment with basic substance on a kind of register of applications, and we can apply it even a few days before harvest. Do not take care of residues because they are compounds that we already eat usually. So today, 3 research teams will update us on running studies on those basic substances. And let's try with Silvia Toffolatti, that she's professor at University of Milano in Italy, And her talk is titled Potential Basic Stance for the Control of Grapevine Downy Mildew and Potato Leaf Blight. Silvia, the floor is yours. You can unmute yourself.
Okay. Okay.
Wow, very nice.
My phone, I think. Okay, you should see my presentation now. Thanks, Professor Romanazzi, for the kind invitation to talk about the researches that we are doing here at the University of Milan. My name is Silvia Laura Toffolatti, and I mainly work on grapevine downy mildew, which is a disease caused by anomycete, Plasmopara viticola. As you can see here in the slide, in the images, the pathogen can affect both leaves and bunches, creating dramatic yield drops if the pathogen is not controlled correctly. Not only the pathogen can directly attack the bunches and create a yield drop, but it can also reduce the quality of the bunches by reducing the photosynthetic tissues by attacking the leaves. Moreover, in case the pathogen is not controlled and the disease is left on its, the pathogen is left on its own, it can also cause an early defoliation of the plant, as you can see on the bottom right of the images. And in here you can see a picture taken in July, at the beginning of July. And as you can see, as you can imagine, there are not any more bunches of this left on this plant. And moreover, the plant is going to suffer during winter because, of course, it's not taking the resources that it's going to need to survive winter. So the pathogen is, on the long term, also causing the death of the plant. And this is a very important impact on grapevine production, because replacing a plant, it's going to cost money for replacing the plant itself, but also it's going to make a waste of time, because the plant needs years to be productive. So for the farmers, it's a very important disease, this one we are talking about. In this talk, I will actually introduce you how we discover the new potential basic substances. And we discover them by investigating the susceptibility levels of the plant towards the pathogen. So we started with a basic study on plant-pathogen interaction to find out, to discover new molecules that can be used for the control of the pathogen, and that can be hopefully let's say, registered as basic substances. So Plasmopara viticola, the oomycete, is not native from Europe, but it's native to North America. And it was introduced into Europe at the end of the 19th century. And from Europe, it spread all over the world with infected grapevine plants. Normally, the resistant germplasm of grapevine is found in the native host of the pathogen, so in North America, and also in Asia, where we have sources of resistance, probably due to the coevolution of the grapevine plants with a cousin, let's say, of Plasmopara viticola, a member of Plasmopara genus. Vitis vinifera, which is the Eurasian grapevine species that is cultivated worldwide for fresh fruit and wine production, is Vitis vinifera, belongs to the Vitis vinifera species. Vitis vinifera is commonly considered as a susceptible species. And here you can see in these beautiful pictures what happens when we inoculate Plasmopara viticola on a susceptible species. And here you can see that the pathogen is developing intercellularly in the leaf tissues. And what happens when we inoculate Plasmopara viticola on resistant American germplasm. Here you can see that the pathogen cannot colonize the tissues, or when it does it, the disease is reduced. The Asian species are in between the susceptible and extremely resistant phenotype. This is what we know from the common information. because normally we cultivate the Vitis vinifera cultivar varieties which are very standardized. We cultivate the same grapevine cultivars all over the world. And we paid little attention to the unknown or less common germplasm. In our studies, started, let's say, 10 years ago, we decided to focus on a particular grapevine germplasm that is located in Georgia, South Caucasus, where the Vitis vinifera variability is very high. The genetic variability of the plant here is very high because they reproduce plant by seeds still nowadays. So we thought that maybe in Georgia, where the genetic variability is high, we could also find new sources of resistance. So we started a project in collaboration with researchers from Georgia, and in particular David Magrazze, and we screened a large collection of grapevine species coming from Georgia, which is present here in Lombardy, in Ricca Gioia. We screened 94 Georgian accessions for disease incidence on leaves and on bunches for 4 years, starting from 2012 and 2015. And we discovered that the disease incidence on leaves and bunches in grapevine Georgian accessions was always lower than a standard Vitis vinifera cultivar, which is Croatina, that is cultivated here in Lombardy. So we moved to the lab and we carried out experimental inoculations where we evaluated the disease severity of grapevine downy mildew, and we discovered that there was a particular accession, L22A, which is called Galoblishvili, which was always getting very good results in terms of resistance to Plasmopara viticola, and indeed also possessed a very low sporangia presence on the leaf discs. So we decided to, and here it's a beautiful picture taken by David Magrazze on Megaloblishvili, which is, as you can see, a black cultivar. So we moved on. We tried to discover which was the resistance mechanism of Megaloblishvili, because it was unique. It was phenotypically different from that we are used to in American germplasm. Here you can see the phenotype that we achieve when we inoculate Plasmopara viticola bianca, which is an interspecific hybrid harboring the American QTLs. And as you can see, when you inoculate Plasmopara viticola, the hypersensitive response is seen as a necrotic response. So the pathogen is not developing. If it develops, it's very low disease severity. With a very low disease severity. In Megaloblishvili, we didn't see any sign of hypersensitive response, but what we see constantly is a reduction in the surface harboring sporulations, so a reduction in the disease severity compared to the susceptible reference Pinot Noir. Here you can see in these confocal microscopy images what happens when we inoculate Plasmopara viticola on Megaloblishvili from 1 day to 6 days after inoculation. And as you can see, the development of the pathogen is similar to that on Pinot Noir until 2 days after inoculation. And we can see that starting from 3 days, the mycelial development is hampered. And at the end of the incubation period, actually at the latency period at 6 days, you can see that the hyphae are completely degenerated in Megaloblishvili. So we see poor sporulation and poor pathogen development. To investigate which was the mechanism of resistance, we undertook a comparative transcriptome analysis where we compared the transcriptome of Pinot Noir, Megaloblishvili and Bianca, inoculated and not inoculated leaves, actually. And what we saw, it was that Megaloblishvili possessed a unique mechanism of resistance, which is completely different from the American mechanism of resistance. And in particular, we saw that in the resistance response, there was an overexpression of some genes that were involved in the terpene biosynthesis, this cytochrome P450 and geraniol 8 hydroxylase. Sorry. So we thought that terpenes could be involved in the resistance mechanism of the plant to Plasmopara viticola. Then we moved on by investigating the VOX, the volatile organic compounds produced by Megaloblishvili, to check if terpenes were indeed involved in the resistance mechanism of the plant. So we undertook a study, and here you can see the reference to the paper if you're interested in. We undertook a study to identify the leaf volatile organic compounds that were produced by the plant, Megaloblishvili and Bianca, upon pathogen inoculation. And then we also investigated the expression of several terpene synthesis genes by real-time reverse transcriptase PCR. So we had, for each plant, for each cultivar, we took 3 plants, and for each plant we examined 4 leaves. 3 leaves were detached and inoculated with Plasmopara reticola, and 1 leaf from each plant was not inoculated with Plasmopara reticola. We placed these leaves in petri dishes, and then we inoculated them and collected the samples at 0, 1, 2, and 3 days post-inoculation. For the analysis of the volatile compounds and gene expression analysis, we kept the samples at -80. The VOC analysis allowed us to identify that 33 VOCs that were produced by Bianca and Megaloblishvili following Plasmopara reticola inoculation. Looking into the details of these compounds, we saw that Megaloblishvili in particular showed a very high terpene accumulation at 2 days post-inoculation. And as you remember, we see the phenotypic response of the plant to the pathogen at 3 days post-inoculation. This means that the pathogen is already, let's say, killed by the plant by 3 days. So the most interesting response of the plant for us is between 1 and 2 days post-inoculation. So we had a confirmation that terpenes are probably involved in the mechanism of resistance of Megaloblishvili. We saw that for some compounds, we have a very significant increase at 2 days post-inoculation, and this is confirmed also in Bianca for farnesen, or that some other compounds, such as nerolidol, are all constitutively expressed by the plant upon pathogen inoculation, and there is no change during time. The gene expression analysis confirmed that there is a significant increase in the expression of the genes which are involved in the biosynthesis of several terpenes. Here you can see osimen, farnesen, and valensen. And so we decided to test the efficacy of these 4 terpenes against Plasmopara reticola. These terpenes are very interesting for us because these are natural compounds that are naturally produced by the plant to counteract the pathogen. So we tested the efficacy of farnesen, nerolidol, osimen, and valensen against Plasmopara reticola with a common leaf disc assay. We bought the compounds from Sigma, from a retailer, and these are almost a mixture of isomers. And we sprayed the leaf disc with the terpenes, inoculated Plasmopara reticola sporangia, and then we incubated the plates for the petri dishes for 6 days, and then we checked the disease severity. And as you can see in this graph, compared to the controls, which are water and water plus 0.2% DMSO, because these terpenes are not water soluble, so we had to check also the presence if DMSO was inhibiting the pathogen growth. And as you can see, at all the concentration tested from 0.01 to 5 grams per liter, all the terpenes inhibited significantly the disease severity caused by the pathogen on grapevine leaf disc. And the protection was the same at all the concentration. And this is good because, as you can see in this picture of the leaf discs that were inoculated with Plasmopara reticola, at the highest terpene concentration, we can also observe some phytotoxicity. So it's good that we have already a good inhibiting activity of these compounds at the lowest concentration. Moreover, we investigated the effect of these terpenes against the pathogen structures by staining the pathogen with cotton blue dye, which is aniline blue dye, a dye which is commonly used for fluorescence microscopy. But since we did this work during the lockdown conditions and we couldn't access the microscopy facilities, we had to switch to an optical microscope assay. So we adapted cotton blue dye to light microscopy. So we developed a protocol with the cotton blue dye, and we observed the pathogen structures under optical microscope, a microscope that we commonly routinely have in our labs. And as you can see in the untreated samples, so the water control, the pathogen hyphae, which are colored in dark blue in this case, are regularly developing in between the cells and are developing also the eustoria, these small dots for nutrition. So here is an eustoria. Then outside from the leaf disc, the pathogen is emitting its sporangiophore bearing with regular branchlets and sterigmata, where the sporangia are differentiated. In the treated samples, the ones which we treated with the terpenes, we always saw the same reaction, and we found out that these active substances all modify, as you can see here, the sporangiophores, because the sporangiophore are not regularly developed. Usually we see a single sporangiophore with branching, but here you can see an hyperbranched sporangiophore, which is sterile. You cannot see the regular branches and the branchlets and also the sterigmata, which are developed at the end of each branch. But here you can see just an emerging sporangiophore, which is hyperbranched, probably also dead, because here you can see all these vacuoles, blue vacuoles, and no sporangiophores. So probably one of the effects of these volatile compounds is to hamper also the sporangia production by the pathogen. So we checked the properties of these terpenes, and these are all sesquiterpenes apart from ocimene, which is a monoterpene. More or less all of them are reported to have an antifungal activity. Some of them, such as ocimene, but also farnesen, are also used in medicine because they have an activity against fungal diseases of humans. And they are interesting because they are active also against mycotoxigenic fungi, such as Aspergillus ocraceus and Aspergillus niger. Farnesen is interesting also because it's used for biodiesel production. And it's interesting since it's so economically relevant. It is also produced by genetically engineered strains of bacteria or fungi, so it can be easily produced in large amounts, which is something that we need if we want to use these compounds. It is not carcinogenic to humans, while ocimene is not cytotoxic at the concentration that we already saw in our slides. Then we check nerolidol. Nerolidol is used as a flavoring agent for animal food, but it's not approved under the regulation. So I don't know if it's going to be interesting to go on with this molecule, because I didn't find any report saying that it's toxic, but it's just written on the EU pesticide database that it's not approved. I don't know why, but it's not approved. So we can evaluate if going on with the nerolidol or not. Interestingly, it's used as a biopesticide versus spider mites in the US. Then we have the last molecule, which is valensen. Valensen is active against Phytophthora ramorum, the agent of sudden oak death. And it's not reported to be genotoxic, so maybe it's an interesting candidate as a basic substance. And it can be also easily extracted from oranges. So this is another good thing, because we can also think about a circular economy approach to achieve this molecule and in a great amount and use them in the field if we need them. What we are doing now on these molecules is to make a dose response analysis to evaluate the minimum inhibitory concentration, and also we are carrying out some blend testing. So we are mixing these terpenes, because we saw that often these terpenes are not used alone but in mixtures. So we have commercial products, plant protection products, which have 3 terpenes together against Botrytis cinerea, for example. So maybe it's going to be interesting to test them in blend and find out if we can reduce the concentration of each active substance to have a good control of the pathogen. Then we are going to program greenhouse test once we have the right concentration of these mixtures or single compounds. And we plan to do efficacy assays in the field. And for this, probably a formulation will be needed, because these compounds are highly volatile and are not soluble in water, so they have these difficulties. So formulation will be probably needed. And what will be very important to evaluate is how to place these molecules inside a disease management schedule, because probably they will be a part of a disease management schedule that is already predetermined. And then we are also running tests on other phytopathogens. Then I want to show you the preliminary results that we are obtaining on working on Phytophthora infestans, which is the potato and tomato late blight agent. In this case, of course, the control is always made of cultural practices, but mainly we carry out fungicide treatments to the fall, to the leaves. What is important of Phytophthora infestans is that it is a cultivable pathogen. So it's different from Plasmopara viticola. Plasmopara viticola is an obligate parasite of grapevine, so we cannot cultivate it on media. We always have to propagate it on fresh, healthy tissues of grapevine. In this case, everything is simple because we can cultivate it in axenic conditions. So Demetrio Marciano, here is his picture, is a PhD student in my lab working on this project on basic substance evaluation and started his PhD last year. One of the interesting things that he found out is that pectin was used by Hill and collaborators to test its inhibiting activity against sporangia germination of Phytophthora infestans. And they saw that already starting from 0.1% weight volume pectin, they had a very interesting response. So they had a very good inhibition of direct and indirect germination of Phytophthora infestans sporangia. What is interesting about pectin is that it is a food additive, and food additives, for their definition, are considered food. And food can be considered as a basic substance. So pectin can be a very good candidate as a basic substance. Actually, it is. If we prove its efficacy against Phytophthora infestans, we can say that it is a basic substance, more or less. So we tested the efficacy of pectin against Phytophthora infestans in this case, and we used an approach which is completely different. We use a liquid medium, and we measured the absorbance to measure the growth of the pathogen. So we inoculated these 96 wells with the liquid medium amended with 0.1% pectin, and we inoculated the wells with a sporangia suspension. And for this, we used a CBS strain of Phytophthora infestans isolated from tomato. Then we immediately measured the absorbance at time 0, and then we measured the absorbance at regular time points up to 4 days post-inoculation. In the end, based on the values of absorbance of the untreated control and the treated wells, we were able to calculate the percentage growth inhibition compared to the untreated control, of course. And what we saw is that indeed pectin had a very good inhibition of Phytophthora infestans sporangia germination and mycelial growth, as you can see here. We had a 80%, 81% almost inhibition of the growth, which is which is of course different from that of a standard fungicide, which we use, like mandipropamide, a cellulose synthase inhibitor. So they are significantly different, these values, but actually we still have an 80% inhibition, so it's very high for us. So we are very, very confident that pectin can be a good candidate as an inhibitor of Phytophthora infestans. And the result was confirmed also at 4 days after inoculation. So now we are of course going on with the investigations on pectin. We are evaluating the activity of pectin on mycelial growth. We are going to evaluate the activity and persistence of this pectin substance against Phytophthora infestans on tomato leaves. And we are also evaluating the efficacy on other phytopathogenic fungi and oomycetes, such as Plasmopara viticola. I would like to thank all the people working on this project, which is very, very wide, because we started from grapevine plants with Gabriella De Lorensis, which made all the studies on RNA-seq and gene expression analysis, Daniela Fracassetti, which carried out the AVOC analysis, Giuliana Maddalena, postdoc student in my lab, which is taking care of the plant pathogen interaction concerning grapevine and downy mildew. And then Demetrio Marciano, the PhD student that is evaluating all the potential basic substances. And then all the other people participating to the project, so the bioinformaticians and so on. And I thank you for your attention.
Thank you, Silvia, for the excellent presentation and for the very good data that you shared with us. For the people that does not know, one of the students of Silvia's lab recently won an award for a video on Phytophthora infestans and grapevine downy mildew. I assume that several questions are ready for you, Silvia. Who wants to start? I will start from one by myself. In the meantime, you can raise the hands. Do you think it's very interesting, this activity of terpenes, because they are not well characterized and studied, but I think there is a high potential about their use. Do you think they can have just an antimicrobial activity, or they can also induce resistance in the host tissues?
I don't know in the specific case of our active compounds, because I didn't check it, but it can happen that the terpenes can induce also resistance. So it's something that we can evaluate actually and can think of. But I don't know in this specific case actually. What we saw is during our transcriptome analysis was that Megaloglishvili seems to have a systemic acquired resistance mechanism, and maybe it could be also mediated by the VOC emission, because maybe.
Thank you. And within this project, Silvia is taking care of the VP-on application of those compounds, while Luis Gonzalez is working, is chairing the VP-on induced resistance. So several skills merged together in this project. Any other questions for Silvia? Or if you want to think a little bit on, and we can open again the discussion at the end of the presentations. So there is from Laura Mugnai, congratulations for the great approach and data. Do you have infection of the persistent of those possible basic compounds?
So for the terpenes, we are sure that their effect lasts until 10 to 12 days post-inoculation. For pectins, no, because we just did a preliminary assay, but we saw that until 4 days, even if the average is quite reducing, but with the standard deviation values, we still are confident that until 4 days post-inoculation, it's still the effect is still visible. We have to see further time points. Thank
you, Silvia. And the further question can be raised to Silvia after the last presentation. You can also write into chat as well for the one from Xiaomeng Guo from South China Agriculture University that will have a talk titled Development and Improvement on Lychee Preservation Technology. Xiaomeng, the floor is yours. You can open as full screen. Perfect.
Okay, thank you for inviting me to give this talk. I'd like to share our recent experience in development and improvement of lychee preservation technology today. Lychee is a subtropical to tropical fruit. The latest research confirms it is indigenous of Yunnan province of China. Lychee is now grown commercially and production in many countries. For example, China, Pakistan, India, Vietnam, Thailand, Indonesia, Israel, Madagascar, Australia, South Africa, and many other countries. China is the leading lychee producing country in the world. Lychee have different harvest season in different produced areas. China has a large produced area and many carnivores of lychee, so the harvest season in China is longer than other countries. Actually, the main carnivores are harvested in May and June. The main lychee suppliers for Europe over Christmas and New Year, Madagascar and South Africa. Lychee fruits have an attractive red persicup and an edible white to cream semi-translucent area that surrounds the glossy brown seed. The area is grape-like in texture, succulent, sweet to acidic and aromatic. Persicup browning, flavor deterioration and decay are common and important problem of post-harvest lychee fruit. Enzyme catalyzes by polyoxidants and lactase water loose and the pathogen infection have been identified as the main factors inducing persicup browning, strong respiration and consumption of sugars and acid lead to deterioration in flavor. Lychee downy blight, anthracnose disease, and soil roots are common post-harvest diseases. Many studies have researched the preservation of lychee fruit, and many basic substances have been tried to be used in lychee preservation, such as acid, amino acid, coating, plant hormone, gas, enzyme inhibitor, cell control bacteria, and some other substances. The main purposes of using these substances are delay persicup browning, prevent decay, and delay senescence. Currently, the use of most substances is limited to laboratory studies. According to our experience, the use of a single substance generally cannot achieve a good and stable preservation effect. And differences in lychee cultivars often lead to large differences in effects. Zinc oxide is currently one of the 5 zinc compounds recognized as safe substance by the FDA. Zinc oxide in nanoscale have found potential applications in food preservation. Nano zinc oxide is a white insoluble powder. We use surfactant to make it into a suspension. Nano zinc oxide treatment inhibited the growth of this pathogen of lychee in vitro test. Dandan and me have tried different concentrations of nano zinc oxide. Although some concentrations have better inhibitory effect, they also cause persica browning and leave a visible white powder on the persica. So we treated lychee fruit with suitable concentration on nano zinc oxide. The figure shows nano zinc oxide treatment delays the persica browning and decay of lychee. After nano zinc oxide treatment, the content of total phenols, total flavonoid, and total anthocyanins and total antioxidant capacity are higher than that in the control and procaloid treatment persica. After nano zinc oxide treatment, enzyme activities which related to persica browning, such as PPO, POD, and lactase, are lower than that in the control and the procaloid treatment persica. SOD activity is higher than that in the control and the procaloid treatment persica. The mechanism of nano zinc oxide is continued to be studied. We also used a combination of brassinolate and kojic acid to preserve lychee. Brassinolate is a phytohormone that can improve the stress resistance of plants and reduce the occurrence of diseases. Kojic acid is an organic acid produced by aerobic fermentation of microorganisms. It is a safety polyphenol oxidase inhibitor. Our experiments have carried out according to the stable of orthogonal test. The optimum formula of preservative was T4. The T4 preservative formula could inhibit the brown and decay of lychee fruit, maintain the color and isotonic content in the persica, delay the diseases of delay the decrease of total soluble solid and vitamin C content in area, reduce the relative conductivity, pH value, and MDA content of the persica, and inhibit the activities of PPO, POD, and lactase. Nearly 1,500 years ago, lychee was a favorite fruit for Yang Guifei. Yang Guifei was beloved consort of Emperor Xuanzong of Tang Dynasty. She was known as one of the 4 beauties of ancient China. It is that lychee fruit are put in bamboo tubes, then the bamboo tubes filled with wax, delivered by the imperial couriers, fast horses, whose riders would take shift day and night to the capital. Of course, the development of preservation and logistic technology make fresh lychee fruit available to people all over the world. This figure reveals the post-harvest treatment of lychee in Chinese different sales models with the repeat development of logistic and transportation. Lychee cells generally only require pre-cooling and low temperature transportation in China. And this figure shows post-harvest treatment of lychee exported to different countries and regions. However, lychee sold abroad take a long time to reach consumers due to inspection and transportation. Except for pre-cooling and low temperature storage and transportation, lychee exported to Australia and Japan need vapor heat treatment. Lychee export to America and Australia need sulfur dioxide fumigation. Vapor heat treatment and sulfur dioxide fumigation are also often combined with diluted hydrochloric acid treatment. The lychee industry commercially uses sulfur dioxide fumigation to overcome the persica browning and the infection by post-harvest pathogens. Sulfur dioxide fumigation imparts a yellow color to the fruit, just like this picture. The origin red color of the persica is regained in a matter of weeks during storage. Dipping in diluted hydrochloric acid can help sulfur dioxide fumigated fruit repeatedly to restore the red color. One of the main constants with sulfur dioxide fumigation is that it leaves undesirable residues. At present, the strict standards enforced on fruit imports by the European Community permits a maximum concentration of sulfur residue levels of only 10 ppm in the edible portion of the fruit. For reducing sulfate residue, we added desulfurization after sulfur fumigation. Control was treated by prochloride. SF was sulfur dioxide fumigation. SF and DS was sulfur dioxide fumigation and desulfurization, then store at 4 degrees. We can say desulfurization treatment accelerated the color recovery of sulfur dioxide fumigated lychee fruit, achieved effect similar to sulfur dioxide fumigation on controlling root and fresh keeping, and reduced the sulfate residue in the sulfur dioxide fumigated lychee so as to ensure its edible safety. There are still many difficulties in lychee preservation technology. We will make more efforts to our researchers so that everyone can enjoy fresh lychee. Thanks the College of Horticulture, South China Agriculture University, China Lychee and Longan Research System, and European Phytosanitary Research coordination support our researchers. I appreciate this opportunity to share and communicate with you all. Thank you very much.
Thank you, Xiaomeng, for the very interesting presentation. And we knew something that we ignored about the lychee growing and storage, because we like to eat, but we don't know how to grow. There is any question from all the people in the audience? I'll start with one by myself. You talked about the vapor treatment of lychee soon after picking. which kind of treatment do you apply? If there is any kind in the vapor of compound, or it is just humid water, or if there is a special concentration of temperature?
You mean vapor heat treatment?
Yeah.
How you mean how to do it? Actually, I'm not sure. Because now we want to find some machine can do these things. We do this because it can change the change the Percy cup, and then if we use some other treatment, it will easy. And any other questions about the vapor heat treatment?
Any other questions by the people in the audience? It does not seem now, but can come after the other presentation. We can open the debate, and further questions can come. So thank you, Xiaomeng. Now is the turn of Benur Matthews and José Pincetta, that will have a talk titled Natural Coatings Combined with Essential Oils for the Control of Anthracnose in Avocado. Welcome. Who is, I think José is starting to present, right? Okay, Benur.
Yes, that's right.
That's right. Okay, thank you, Gianfranco, and good afternoon, everyone. This is a new research line that we are working on in our lab. So we are going to show you the initial results of our research. So thank you for inviting me. And I'd like to introduce you my colleague, Dr. José Sidinaldo Pincetta Jr., that will talk about it.
Thank you, Benur. Welcome, José. You can share the screen.
Okay. Can you see my screen?
Yeah.
Okay. All right. Thank you for the opportunity to present this work that we're conducting in our lab. Thank you for Professor Benur to invite me to give this talk as well. So we are going to show you some initial results that we have from our lab, and this talk is entitled Natural Coatings Combined with Essential Oils on the Control of Anthracnose in Avocado. So just to give you an overview, the avocado is a fruit native to Mexico and Central America. This is a fruit that has high nutritional values. It's rich in unsaturated lipids, vitamins, and fibers. Brazil is the sixth worldwide avocado producer, and only 10% is currently exported. So mostly the Hass variety is exported. So the last data that we have shows that in 2019, Brazil produced 243,000 tons of avocado, and only 10% was exported. In terms of the country, São Paulo state is the state that most produced the avocado. 51% of the national production is in the state. The main varieties that we plant in Brazil are these ones. The Hass variety is the one that is the most new in the country. It's recently the production. We are most used with these varieties that are with a sweet taste. So just for as a curiosity, in Brazil, we are used to eat avocado as a sweet fruit. instead of many countries around the world that is mostly eaten as a salty or in salads or in other dishes. So these varieties are rich in sugar and less oil content. So we are most used to these varieties. And Haas variety is quite new in Brazil. And a few research is done with this variety. So anthracnosis is the most common disease in the post-harvest of avocado. So here we have some pictures of the main symptoms of this disease in avocado. And in our research, we conduct some in vitro experiments for screening and assessing some species with potential inhibitory capacity for the anthracnose disease, the anthracnose agent. So we conduct some tests, and we find out that these three species, Lipiacidoides, Thymus vulgaris, and Symbopogon citratus, were the ones that gave the lower minimum inhibitory concentration needed for the assessment. So we used these three varieties, three essential oils to assess their effects in vitro. So we used these concentrations for thyme essential oil. For lemongrass, we use these concentrations, and for pepper rosemary, we use this concentration. Here we have a picture of the pepper rosemary Lipiacidoides, which is a plant native to Brazil. So we inoculated 5 millimeter discs of the mycelium in petri dishes with BDA plus the essential oils in the concentrations that I just showed you. And for the in vivo experiments, we use the Haas avocados harvested at commercial productions. The fruit were inoculated with 25 microliters of aconidial suspension. And the treatment consisted of a control with carnauba wax, and the combination of carnauba wax with the essential oils. So we tried to understand, we tried to join and combine the benefits of the carnauba wax, that is a widely used wax for coating the fruit here in Brazil. And we tried to see the benefits that the addition of essential oils to the carnauba wax would result in the fruit conservation and inhibition of the disease. So we tested these concentrations with Lipiacidoides and these concentrations with lemongrass essential oil. The product was stored for 14 days at 12 degrees, simulating refrigerated storage. And the second evaluation was done after 3 days at 22 degrees, simulating a condition in the shelf. So we have some results that we obtained for the in vitro tests. With the thyme oil, we have that the minimal concentration for a total inhibition was 200 microliters per liter. So here we have some graphs as well, showing that the thyme oil at 200 microliters could inhibit 100% the mycelial growth, and also the conidial germination. Here we have the result for Lipiacidoides oil. So the minimal concentration also was 200 microliters. this concentration could inhibit mycelial growth and also conidial germination. And for the lemongrass oil, the minimal concentration needed was 1,000 microliters per liter. And here we have also the mycelial growth inhibition, and for conidial germination, the best results was with 200 microliters per liter. In our in vivo results, also we checked the lesion diameter and the disease incidence. So for the lesion diameter, we could see that at 17 days of storage, the treatments with the essential time essential oil could could inhibit the lesion diameter or the severity of the disease. Also for the elicidoids, after 17 days of storage, the treatments with 200 and 1,000 could inhibit the severity of the disease and also the disease incidence. For the lemongrass oil, the results were the lesion diameter was inhibited, or the severity of disease was inhibited at the concentration of 2,000 microliters per liter. Also, we assessed some enzyme activities, as we suppose that the essential oils and the modifications that the coating do in the fruit can trigger some responses related to the metabolism of the fruit. So PAO activity was influenced by the coating with thyme oil and carnauba wax at 17 days of storage, and also the lemongrass oil could inhibit the PAO activity. For the pod activity, we had that all the concentrations that we test for thyme oil was able to inhibit the activity of the POD enzyme. Also for the elicidoids, we also had some inhibition of the PAO activity after 17 days of storage. For the ethylene production, the most significant results we obtained was with thyme essential oil. The thyme essential oil could inhibit the production of ethylene in all the days that we assessed. For elicidoids and lemongrass, the results were not very promising in terms of ethylene production inhibition. And for the respiratory rate, we had some good results with the thyme essential oil and elicidoids essential oil that could inhibit the respiratory rate promoted by the combination of the coating with carnauba wax and the essential oil. Our preliminary conclusions are that the mycelial growth and conidial germination can be progressively inhibited with the increasing concentration of essential oils, and that the highest concentrations of essential oils with carnauba wax could inhibit the severity of the disease showed through the lesion diameter. Thank you very much for your attention, and we are happy to answer any questions that might have. Thank you for the opportunity, Professor Gianfranco.
Thanks to you, Jose and Benu, for the very great results that you showed. Very, very interesting, a lot of trials and a lot of information. There is any of the people attending the webinar that wants to have questions to Jose or to Benu? I'll start with one by myself. did you find any phytotoxic activity by any of essential oils? Alessandra wants to raise the same questions, maybe.
Yeah, yes, not so far. We did it with this preliminary result. If with these tests in the in vivo tests, we couldn't find any visual toxicity effect for these oils that we tested.
Okay, so the overall mechanism of action of this essential oil, which one you can predict that is?
The most promising, based on the general results that we have, is the thyme oil.
Yeah, hello, Jose, and thanks for the nice presentation.
Thank you.
Maybe this can that you can find any kind of phytotoxicity, but maybe because use the carnauba wax.
No.
Combined effect.
The combined effect. Maybe not, because at the treatment we used a control with carnauba wax, right? So we also didn't see this effect in the control, in the water, in the control. So maybe this is a possibility of the carnauba wax have a combined effect. Yeah.
Yeah. I don't know very well the effect of thyme essential oil on avocado, but I know the effect of essential oil of thyme on maybe on apples and other kinds of fruits. And yeah, high values of this essential oil are very destructive on fruits, and you can see sometimes phytotoxicity.
Yes, that's a good point that you raise, because we didn't test exactly only the essential oil on the fruit, so maybe this is a combined effect that result in this non-phytotoxicity. Yes, this requires first investigation. Thank you for pointing that out.
Thank you.
Simona Saltani.
Congratulations for a nice presentation, and just a curiosity, if you are planning to conduct a test with combinations, I don't know, maybe also this could, using a combination of essential oil could reduce the concentration, so maybe prevent toxicity phenomena. And also, if you are planning also to perform the enzymatic assays a little bit earlier, maybe you could see stronger effect as compared, because as I saw, you tested 14 and 17 days.
Yes, thank you for your question. We are planning to do more combinations in terms of essential oils and other types of coatings. For example, we planning to do a combination with chitosan and another edible coating that we have been testing in our lab, but we don't have many results so far. So we are planning to do another combinations. In terms of the enzymatic activity, that's a good point for us to analyze an earlier point of assessing the enzymatic activity, because maybe it was too late the point that we evaluated. This is a good questions to address in our future experiments.
Thank you, Jose. And now there is a question that is for all three speakers. Let's start with Sylvia about the formulation of those compounds. So starting from terpenes and later to Xiaomeng and then to Jose. Sylvia, what about how far we are from formulation, and if there is the chance to formulate, if you are thinking about which are the main difficulties and so on?
Formulation, I think it's the most important stage now, because finding effective substance is quite easy, more or less, but then making them available on the field, it's going to be very tough, because volatile compounds are volatile, so they are not going to persist. They are not soluble in water, so you need to make them soluble in something. And so this is tricky. But the good thing is that there are some patented techniques to micro-encapsulate these essential oils in microspheres that can release the active substances in presence of water. So this is very good for Plasmopara viticola, because we know that the pathogen is infecting by food, and to infect it needs water, because the zoospores need to swim in water. So when there is water, these microspheres are going to release the active ingredients, so they are going to inhibit the infection process. This is something which is patented, so I don't know if it's going for all the people how easy it would be to use these terpenes directly. We cannot think, I think, to use them directly. We have to formulate them. We need a specialized people which is specialized in formulations, because formulations are very important for a plant protection product. So I don't think that in my case that these terpenes are going to work in the field like this.
Oh, tomorrow. Xiaomeng, thank you, Silvia. Xiaomeng, which is your experience? How far you can be from a potential of the solution that you use, application and formulation? Can you unmute yourself, Xiaomeng?
Sorry, can you repeat the question?
Yes, the question was, do you think to a possible formulation of the solution, of the most promising solution that you applied, a commercial formulation? You're not forced.
I'm not. Because lychee fruit, I think it's very, the pericarp is very thin, and the cultivars is very. We have many different cultivars of lychee fruit, and different cultivars, different formulations. So I think because we tried the same concentration, same substance on the lychee fruit, but the cultivars is different. Large differences between the cultivars. So I think no formulation. That is why I will cite the substance only used in the laboratory and in the commercial. We use sulfur dioxide fumigation and vapor heat treatment. We want to change because sulfur dioxide fumigation have some problem, but we don't find a better substance to change it. So I think we have a long road to we have many difficulties to solve this problem. So I think no formulation. We must try.
Thank you, Xiaomeng.
Thank you.
Thank you. It's very ambitious to replace sulfur dioxide, but one day we may find something that can be equally effective. The same question for Benur or for Jose. Who wants to answer? Do you think we can move toward something that can be applied on large scale?
I think so. I think that we could do more. It requires more fine-tuned experiments for sure. But I think a possible commercial product is potentially has a potential to be obtained. In Brazil, we have a widely use of the carnauba wax already. So introducing the essential oil as well as we need to study as well how to maintain this essential oil in the product, right? We need also to carry these studies. But I think that has a great potential to become a solution for commercial production.
Thank you, Jose. Simona?
No, not a question, just a consideration that probably post-harvest environment is particularly suitable for the kind of products you are proposing, all of you today. So very interesting. Thank you.
Thank you to you. Any additional questions? If not, I thank all the three speakers and all the people that followed the webinar. It was very interesting. So if somebody missed the first part and want to see again, we will upload it on the YouTube channel. And we are planning a next one on 15 of March at the same time. So we will have other novelties on use of basic substances and potential basic substances. So thanks for being all together today, and see you next time. Enjoy, have a nice day, have a nice time. Bye to you all.